We've been talking about translation, and so I wanted to do one more thing to try to put the concepts together. Here we have all of the parties that are involved in translation. The ribosome, which is made out of ribosomal RNA and protein, is depicted here as a large subunit and a small subunit. The messenger RNA is depicted here by these letters. You can tell it's RNA because it's got G's, A's, and U's and C's, right? And the messenger RNA, this is the complement of one of our genes after it has been spliced and sent out of the nucleus. These are the instructions for this is the protein or peptide that we're supposed to make today. We're going to act as the ribosome. How do we know how to make the protein? Well, we don't have to know, because these guys here that look like little rainbow things, those are our transfer RNAs. I want you to notice that each transfer RNA is carrying a 3-letter amino acid. This one says capital T, lowercase h, lowercase r. That's threonine, and that's one of the amino acids. Now, threonine is an amino acid, and the codon for threonine is ACC, right? You don't have to memorize that at all. I haven't memorized that. How did I know that? I know that the codon for threonine is ACC because the anticodon here in our transfer RNA is UGG, and UGG would be the complement for ACC, right? So here's what's going to happen. Every protein that's being made starts with a start codon. The start codon is AUG. So I have to find the transfer RNA that has the anticodon that is the complement for AUG, which is, of course, UAC. Once I do that, then the small subunit of the ribosome will join, and then the large subunit of the ribosome will join, and now the next transfer RNA will come in. Which transfer RNA should I grab? Well, here's the codon CCA, so what do I need? I need GGU. Why? Because GGU is the complement to CCA. It is the match, right? Now, it turns out that the transfer RNA that has the anticodon GGU carries a proline. That tells me that CCA is a codon for the amino acid proline. And what happened while I was busy talking was that the ribosome created a peptide bond between the first amino acid methionine and the second amino acid proline. And then the first transfer RNA no longer had its methionine attached to it, and it was free to go, and it's welcome to come back later on. So let's go ahead and build our protein. This is the codon, GAG. What is it the codon for? Ribosome doesn't know. I don't need to know. I do need to know that the complement for GAG is CUC, and I need to grab that transfer RNA. And I can trust that that transfer RNA is carrying the proper amino acid, which is glutamate in this case, right? By the way, a mutation that would make it so that a transfer RNA is not carrying the proper amino acid would be so dire that it never creates a living organism. So you will never see that mutation in life because it kills every cell that has it. So ACC is our codon. We're looking for UGG. UGG was our first one, right? We'll grab UGG. Great. Now we've got a little peptide, 1, 2, 3, 4 amino acids long. We're ready for GCA. GCA is the anticodon for the transfer RNA. Now we've got arginine. Now we need UCC. This is interesting. UCC, that also codes for arginine. Isn't that interesting? We're going to get to that later in this lecture or maybe the next video. Now we need UUU because our codon is AAA, so we need the anticodon UUU. Do you see how the ribosome, is it reading the mRNA one codon at a time? Yes, but it's not actually translating it. It's really just going, okay, I need a UCG here. All right, here comes UCG. And UCG is just carrying the proper amino acid. Now we need UAC. Then we need valine, CAA, good. And then we get to UAA, and UAA, that is a stop codon. So that tells this ribosome, we're done. You can move on and start over again. Okay, I hope that cleared things up a little bit. So we have come to this summary that DNA transcription takes the DNA blueprint and it makes an RNA version called the messenger RNA. It makes the complement of the gene. Now, that first RNA version is not actually the final messenger RNA. It is pre-messenger RNA. And pre-messenger RNA is made out of introns and exons, and it needs to be spliced. And because that pre-messenger RNA in splicing can be put together in different combinations, that allows a single gene to create the instructions for many different proteins. The messenger RNA leaves the nucleus through a little nuclear pore, and then the ribosomes are going to just do whatever they're told, right? Ribosomes are very cooperative workers. Whatever instructions they get, that's what they build. And the mRNA is a series of codons, and it gets used by the ribosome to make the proper protein, with the transfer RNAs bringing the amino acids one at a time into the ribosomes. Now, there was one more thing I wanted to tell you about why this thing, molecular biology, is such a brilliant solution to the problems that life has. Getting an amino acid wrong can be dire. So this is the case of sickle cell anemia. In sickle cell anemia, the DNA is wrong by a single nucleotide. Instead of a thymine here in this codon, it is an adenine, okay? So it is wrong by a single nucleotide. And that single nucleotide will make this codon wrong by a single nucleotide. And that is enough to change a single amino acid in the chain. Now, the gene that causes sickle cell anemia is a gene for one of the subunits of a protein, hemoglobin. And hemoglobin really fills up your red blood cells, and it's the protein that makes your blood look red. That protein subunit is more than 100 amino acids wrong, but getting just one of them wrong will create the problem of sickle cell anemia. Now, sickle cell anemia, if you take a blood sample from a person with sickle cell anemia, most of the time, their red blood cells look fine. They look very, very normal. It's only when the hemoglobin in the cells of a person with this mutation, it's only when the hemoglobin in their red blood cells are exposed to certain stresses, like extremes of pH or very, very low oxygen, it's only in those situations that their hemoglobin will spontaneously denature. And when it does, it pushes the shape of the normally disc-like red blood cell and turns it into this sickle shape. And that creates a profound problem that in the absence of modern medicine would generally be fatal for the patient. That's how important it is to not get the problem wrong. So let's take a moment to talk about gene mutations. I want you to know two different kinds of gene mutations. Golly, my slide is a little bit messed up. Let me try to move things. Okay. I don't know. All right, let's just go back here. There are two kinds of gene mutations I would like you to know about. First of all, you should know that mutations are changes in the DNA. Okay, that's a mutation. And when there's a change in the DNA, it'll be permanent. Not only there's only a change in the DNA if that mistake made in copying the DNA was not caught and fixed by the proofreading polymerase, right? But if it's not caught and fixed by the proofreading polymerase, then it's in the DNA. And then every time that cell divides and replicates its DNA, well, that mutation will be there forever and ever. So there are two different kinds of mutations. The first one we'll talk about is a frameshift mutation. You should know the definition. It is the insertion or the deletion of a nucleotide from a coding region. Let's imagine that this thing, UUU, of course, this is not DNA, this is messenger RNA. Okay. UUU, that's going to be what our messenger RNA should have said. So up here, UUU is phenylalanine and AAA is lysine. So when we made this peptide, it should have said phenylalanine, lysine, phenylalanine, lysine, phenylalanine, lysine, right? That's what it should have said. With a frameshift error, in this case, one of the nucleotides is gone. It's missing. It got skipped, all right? So now it says UUA, AAU, UUA, AAU. Remember, it was supposed to say phenylalanine, lysine. Now what does it say? Now it says leucine asparagine, okay? We've completely messed things up. A frameshift mutation does not just mess up one codon. A frameshift mutation messes up that codon and generally will shuffle everything after that codon, at least in that particular exon. That is why frameshift mutations generally are catastrophic. They will cause a very large change in the protein that is made, and that often is not survivable. Okay? So let's look at the other kind of mutation. The other kind of mutation is this one here. It is a point mutation. It's called a point mutation because it is a substitution error. So in the frameshift error, I deleted this uracil. But in a substitution mutation, I have just substituted a cytosine for a uracil. Now, I want you to watch this, okay? We have changed this particular codon. This codon should have read UUU, but now it reads UUC, okay? So if we have got this thing called a point mutation, we will have changed one codon, and we only might have changed the amino acid that's going to be put there, and we only might have changed ultimately what the protein does. Why? Because it turns out that UUU as a codon and UUC as a codon, they are both codons for the same amino acid. So that means, for those of you keeping track, that there was a mutation in the DNA. It is a point mutation, and it changed one codon to a different codon, but because it was a point mutation, and it did change one codon, it didn't change the amino acids, so it didn't change the protein at all. Guaranteed that you and I, whoever you are there, that we have got proteins in our bodies that if I look at the protein and its amino acid sequence, they are absolutely identical. Identical. And yet, it is also very likely that your DNA for making that protein is different from mine. For example, in this situation, yours might code for mRNA that says UUU, and mine might code for mRNA that says UUC. Which of us has the gene mutation? Yeah, we don't know, okay? Most of the time, we don't know which one is the mutation. But we do know that the reason that they are different is because of mutations, and point mutations are very survivable, right? We will start there at the beginning of the next video.